AP Biology Unit 1, Intro to Lipids. There are 4 types of lipids: phospholipids, fats and oils, steroids, and waxes. Lipids contain carbon, hydrogen, oxygen, and sometimes phosphates. These molecules are mostly nonpolar, made of carbon-carbon and carbon-hydrogen bonds. They typically have a polar region, which usually contains carbon and oxygen bonds. Lipids can be found in cell membranes and organelle membranes. They're used for insulation and energy storage. They can be hormones, and they can also be used for waterproofing. Fatty acids are long nonpolar tails with a polar head. There are 3 types of fatty acids. Saturated fatty acids have only single bonds and they're straight nonpolar carbon chain. You can notice the repeating carbon-carbon-carbon bonds, with the very end being a carboxyl group. A cis unsaturated fatty acid has a double bond that contains a single hydrogen on the same side of the double bond. This causes a bend in the nonpolar chain. For example, look at this cis double bond here. Both hydrogens are on this side of the double bond. This makes a large kink or bend in the structure. A trans unsaturated fatty acid has a double bond that contains the single hydrogens on opposite sides or trans of the double bond. This causes less of a bend. Notice the nice long straight chain here with a slight bend in it, and then it continues on. The hydrogen is on opposite ends of that double bond. Lipids. Fats. Fats consist of a glycerol molecule and 3 fatty acids. The fatty acids are added to the glycerol through a dehydration synthesis process. A hydroxyl group from the glycerol is combined with a hydroxyl group from the fatty acid. Here you can see that the hydrogen is stripped off of the glycerol, the hydroxyl is stripped off of the fatty acid, and a water molecule results. This is called an ester bond. You can see these 3 different fatty acids have been attached to this glycerol molecule. Note, these fatty acids, they don't have to be the same acid. Fats and oils contain different types of fatty acids. Take a look at this nutrition label from the mayonnaise jar. There's saturated fat, polyunsaturated fat, and monounsaturated fat, all within the same can. Here's another way of looking at a saturated fatty acid. Stearic acid has a long chain of just carbon and hydrogens, all single bonded. Here we have cis oleic acid. Partway through the molecule we have a double bond with the 2 hydrogens on the same side of that double bond. Creates a large bend in the structure. Transoleic acid has a nice long chain, and the double bond here has the hydrogens on opposite sides of that double bond. Note, the kink in this, or the bend in this, isn't as great as the cis oleic acid. How does the type of fatty acids in your ingredients of mayonnaise or butter affect how easily it is spread on bread? Fats are known to be solid at room temperature, while oils are liquid at room temperature. Long straight chains of fatty acids can pack nice and tight together. Oils contain double bonds in their fatty acid chains. This can make a kink or a bend in the fatty acid, and it acts to decrease the density of the oil. Let's look at the following areas on this fat molecule. The first structure here is a nice saturated fatty acid. Notice there are no double bonds in this structure. This also has no double bonds. It is all complete carbon-carbon single bonds, completely saturated with hydrogens. This structure has a double bond in it. The double bond has the hydrogens on the same side. This part of the molecule has 3 ester bonds. We have an unsaturated fatty acid, an unsaturated fatty acid, a cis fatty acid, and this is the glycerol backbone with ester bonds for attaching the fatty acids. Lipids can also be phospholipids. These create membranes. What's different about a phospholipid is that it has a polar head containing a phosphate group. The head of a phospholipid is hydrophilic because the phosphorus, oxygen, carbon-oxygen, and carbon-nitrogen bonds are all polar. These are all molecules that are in this head. If you look up here, you can see the actual elements. We have nitrogen, oxygen, phosphorus, and oxygen. Those all make this section very polar. The tail of this molecule is hydrophobic. You notice it's just made out of carbons and hydrogens. There is a bend in this long hydrophobic tail, which shows that there is a double bond. Long chains of carbon-carbon and carbon-hydrogen bonds, these are all nonpolar, making it hydrophobic. Phospholipids can create a bilayer sheet. These polar heads here face on the outside towards water. The inner side is going to contain the hydrophobic tails. Steroids are also lipids. They contain 4 carbon-based rings and they're fused together. Take a look at this. This is 1 ring, 2, 3, 4. Aldosterone also has 4 rings. 1, 2, 3, 4. The differences have to do with the different functional groups. Other examples of steroids are estrogen, testosterone, cholesterol, and aldosterone. If you take a look at these 3 different steroids, this is cholesterol. Notice that cholesterol has a long hydrophobic tail on it with the 4 fused rings. Testosterone is similar to cholesterol, 4 fused rings with a hydroxyl, methyl, methyl, double bonded oxygen, and a double bond in the ring structure. Estradiol, which is similar to testosterone, has a single bonded hydroxyl group. 3 double bonds here, a methyl and hydroxyl. The difference between testosterone and estradiol isn't that great. Notice in this structure we have phospholipids. We also have some cholesterol molecules that are attached. These cholesterol molecules have, instead of a polar head, they have the small hydroxyl group. And then this very long end, or hydrophobic tail, sticks on the inner surface of the cell membrane. Waxes are also a type of lipid. Look at the difference here. We have long chain of carbon and hydrogen bonds with 1 double bonded oxygen and another oxygen. Waxes assist in creating a protective barrier for lip balm. You can see in this succulent is covered with wax. The wax that this plant creates helps protect it from the harsh, dry environment. Beeswax is used a lot for things like cosmetics, lip balms, and lotions. The long nonpolar hydrophobic tail acts as a barrier to polar molecules like water. Water that is on the lips cannot pass through the tail of this lip balm, and it acts as a protective barrier. Let's get out your modeling kit. Your membrane student modeling kit will help determine or help you visualize how lipids and phospholipids self-assemble. This is a micelle. Notice all the polar heads are on the outer surface, and on the inner surface are the nonpolar tails. The nonpolar tails will automatically face each other. The polar heads also line up next to each other. Notice that this is fluid. These molecules are not bound to each other in a rigid covalent structure. Rather, they are bound together because of their hydrophilic and hydrophobic interactions. If you look at the partially positive hydrogens in water molecules, they will align themselves with this negative polar head that's found in lipids. The water molecules are not going to find their way in between these hydrophobic tails.